Traditionally, animals have been classified by body symmetry, which is dividing an organism on a plane. They also been classified by embryonic tissue types and how the digestive system forms during embryonic development. If you take a look at this cladogram, you'll notice this group called the comb jellies, phylum Ctenophora. They appear to be an outgroup of animals, and although they appear a lot like other jellyfish, which is phylum Cnidaria, they differ genetically. One being is that ctenophorans lack the HOX, H-O-X gene, which is found in all other animal groups except sponges and placozoans. The HOX gene is important in embryological development and explains the similarities in animal embryos up to a certain point during development. Zoologists can divide animals into groups based on body symmetry. Before beginning on symmetry, we need to talk about relative positions in animals. Just like in geography, we can say that one city is north of another city, but that another different city can be south of the original city that we had just mentioned. We can use body positions to describe where you are relative to an organism. So for example, ventral means towards the bottom of the animal. Dorsal means towards the top, and so on and so forth. Symmetry is the ability to divide an organism into two parts by at least one plane. The three planes used to divide animals are the sagittal plane, which divides an animal into equal left and right halves in the middle of the animal, the coronal plane, which divides an animal into top and bottom halves, and the transverse plane, which divides an animal into cranial and caudal halves. Asymmetry, which is found in sponges and placozoans, are animals with no symmetry whatsoever. These body plans have no specific shape, or if they do, it isn't considered true symmetry since these animals lack tissues and organ structures. Radial symmetry is found in cnidarians, or the jellyfish, ctenophorans, also known as comb jellyfish, and echinoderms, which includes sea urchins, sea stars, and the like. These animals are circular or barrel-shaped, leading to limitless forms of symmetry. Most animals are bilateral, and that they can only be divided by the sagittal plane. Most of these animals also have a head region where most nervous functions are located. With the exception of sponges and placozoans, all animals carry out embryological development with germ layers. These tissues give rise to the different body systems found in animals and begins during gastrulation of embryonic development, which is the formation of the gut during embryonic development. Diploblastic animals, such as phylums Cnidaria and Ctenophora, only produce an endoderm and an ectoderm, with a non-living layer in the middle. This non-living layer becomes the jelly layer that you feel when you touch jellyfish. Triploblastic animals make up the remainder of the animal kingdom and have an additional mesoderm layer. Amongst triploblastic animals, they can be divided into three groups based on the presence or absence of a coelom. A coelom is a body cavity or opening in the body that derives from mesoderm tissue and allows organs to grow and move around. In humans, we have several body cavities, such as our thoracic cavity holding our lungs and our heart, abdominal cavity with much of our digestive organs, and cranial cavities for our brain. The coelom is usually filled with fluid to help lubricate the organs as they slide past one another. They provide cushioning and shock absorption, as well as allowing for the exchange of gases, nutrients, and waste via diffusion and osmosis. Bilateral triploblastic animals can be divided into three categories. First are the acoelomates, which lack a coelom altogether. This is only found in flatworms. Eucoelomates have a true coelom that is completely enveloped around mesoderm tissue. Most bilateral animals have this. Pseudocoelomates have a false coelom that is sandwiched in between mesoderms and endoderm tissue. Only roundworms have this. Amongst the bilaterally symmetrical triploblastic eucoelomates, did you get all that? You can further divide them into two groups based on the differences in the origin of the mouth. The digestive system begins in the mouth and ends in the anus. And recall that gastrulation, or the formation of the digestive system, is the first organ system made in animals. The blastopore is the beginning opening in the embryo during gastrulation and will either be made into the mouth or anus depending on the grouping. Protostomes, which include mollusks, roundworms, segmented worms, and arthropods, have a blastopore that develops into the mouth first and hollows out the gut to make the anus last. Deuterostomes, which include echinoderms and chordates, have a blastopore that develops into the anus first and hollows out the gut to make the mouth last. Sorry, guys, that's what we are, chordates. These groups also differ in how the coelom is made. Protostomes go through a process called schizacely, where specific blastomere cells migrate into the interior of the embryo and form the hollow opening as the coelom. Deuterostomes go through enterocele, where mesoderm pounds break off of endoderm tissue and migrate and fuse to make the coelom. Both groups differ in the cleavage of the zygote. This is the notable 8-stage cell development. Protostomes go through spiral cleavage, where the top layer of cells rotates slightly and appears misaligned from the bottom layer. Deuterostomes go through radial cleavage, where the top and bottom layers are aligned. In addition, protostomes have a determinant cleavage, where the role of each embryonic cell is already determined. Deuterostomes have indeterminate cleavage, where the role of each embryonic cell is not determined until later on. In other words, deuterostomes are made of embryonic stem cells until later in development. In our next video, we will be looking at animal phylogeny as well as the evolution of the animal kingdom.